Well, good evening. Thank you very much, Dan, for the kind introduction. Thank you to the organizers for inviting me, as a matter of fact, to the second Cold Spring Harbor meeting in a row. I have been here on this podium also on Friday. So what I will do in the next 35 minutes or so, but certainly not longer than the soccer match between Germany and Portugal will begin, I will tell you a bit about the 26S proteasome. What I will do in the first part is I will give you an update on the structure of the 26S, and I will elaborate a bit on the methods which we used to determine the structure. Then in the next part, I will give you at least a glimpse of the dynamics of this molecular machine, a picture that is still substantially incomplete, but at least I think it will show you the range of the dynamics. And in the last part, I will show you something that is work in progress, and that are structural studies of the 26S proteasome, not in isolation, but in situ, in their intact cellular environment. And for that, I need also to give a little bit of an introduction into cellular electron tomography. So basically, I will in the first part, as I said, elaborate on how we determined the structure, and that is meant to be illustrated in this picture here, which was, designed as a cover, but it was rejected by the editors. So I think the main ingredients in the structure determination was the structure, the EM structure in a frozen hydrated state. And it was, it was interpreted in the light of data which came from cross-linking and mass spectrometry. So that helped quite a bit to guide the interpretation of what was at the time a kind of a medium resolution structure. Now, this has been the textbook image of the 26S proteasome for quite a long time. And it was, I was always frustrated to see it. It was a structure which we did, well, late last century. It was not very informative. Resolution was just too low. Basically, what it showed was, there are two subcomplexes, the 20S proteasome or core complex, and as most of you will know, that is where the proteolysis takes place in a cavity which is formed by the beta subunits here. Then there are the alpha subunits which enclose a gate which provides access to the interior of the 20S core. And at the same time, it's the platform for the docking of the regulatory particles. Their role is basically to prepare substrates for degradation, to recognize ubiquitinated substrates, to deubiquitinate them, to unfold them, and to assist in their translocation into the core complex. Now, the structure of the core complex we had determined almost, basically 20 years ago by X-ray crystallography. So we had a detailed structure for that, but not for the remainder of the complex. Would it be the challenge today or the task today to determine the structure of the 20S? We would probably not do it by X-ray crystallography. We would do it by EM single particle analysis. Just an example here, that is the structure of the same 20S from Arkea. It has been done by single particle EM to the same resolution as we had at the time, 3.67 angstroms by EM. That is the density map, and we have superimposed the X-ray structure. It's a perfect fit. The difference is that was something like 3 years of crystallization trials, and this was, no crystallization and 9 hours of data collection, followed by a day or two of processing. I think that is that development, and that was even done without the technology, the detector technology that became recently available. Now, however, determining the structure, nevertheless, of the 26S is much more challenging. The reason for that is kind of threefold. There's, first of all, the sheer complexity of the 26S, the labile nature, and most important, we'll come back to that, the heterogeneity of these samples. So basically, the complexity, yes, there are 33, 34, what we call canonical subunits. There are the 7 alpha and the 7 beta subunits of the core particle. There are the 6 closely related, nevertheless functionally non-redundant AAA subunits. There are the 2 resident ubiquitin receptors. There's the small thing here, RPN15 or SEM1, which is basically a glue between 2 of the PCI subunits. Then there are the MPN domain containing subunits, two very close homologs, of which only RPN 11 has aminocatalytic function as a ubiquitinase. And there are the 6 related PCI module containing subunits, but different in size. And their role is basically that of a scaffold embracing these 2 subunits here. And then there are the 2 largest subunits, RPN1 and RPN2, which are highly dynamic, as you will see later on, and they provide, docking sites for shuttling or associated subunits, including, for instance, UBP1. Now, as you take a low-resolution image of the 26S done a couple of years ago, and then compare all the molecules in the data set with each other, then you can generate a variance map. The variance map is what is shown in orange here and superimposed to the structure. It highlights where in the structure the differences are. And you see that basically the core particle is more or less invariant. That, of course, facilitated its crystallization. You see that the regulatory particles are extremely variable, and there are 2 sources of heterogeneity. One is compositional, because there is a fair number of subunits that is present in the complex in substoichiometric amounts, including ubiquitin receptors. Plus, there is enormous conformational heterogeneity driven by the motions of the AAA ATPase subunits. Now, I think I will recapitulate briefly, how the structure was determined, and then go into some sort of the refinements. Now, first of all, these are the ingredients, if you like, the EM map. We had to know, but that was kind of trivial, the absolute positions of the AAA ATPase module. We had to determine, that was less trivial, the absolute positions of the 2 ubiquitin receptors, RPN10 and RPN13, because they are located so kind of distally in the complex that we didn't obtain any crosslinks, or at least no trustworthy crosslinks for those 2 subunits. Then a very important element was, basically proteomics networks data obtained by, as I mentioned, cross-linking and well, the sequence-specific cross-links obtained from mass spectrometry, plus increasingly atomic models of some of the subunits. Now, the structure which we used to generate the initial model was an 8 angstrom structure of the POMB 26S proteasome. It was still the data were still collected kind of manually. It was built from, a total of 30,000 electron micrographs, so which took basically 1 PhD lifetime to collect the data. And this led to this structure here, an intermediate resolution structure. Now, the AAA ATPase, basically we knew anyway where it was, but we had to determine the boundaries a bit more accurately. And that was done by taking advantage of the fact that, it has pseudo-hexagonal symmetry, whereas the core has pseudo 7-fold symmetry. So, doing some averaging, rotational averaging, enhances everything that follows the symmetry. That gave a rough outline. We could threshold it. And then we used that as a starting model to refine basically the boundaries of the AAA ATPase, as you see here. It came as a surprise to us, and we didn't believe it initially really, that, the 6-fold axis or the pseudo 6-fold axis of the AAA ATPase and the pseudo 7-fold axis of the core particle were not really aligned, but were sort of, there was an offset, and there was also a certain inclination. I will come back to that again quite a bit later. We also made a proposal at the time for the order of the subunits of the AAA ATPase subunits. We came up with, it was based on published interaction data of a whole range of data, many of them, as a matter of fact, quite contradictory. But the order we determined was 1, 2, 6, 3, 4, 5. And we built then a homology model, and it fitted quite nicely into the boundaries we had determined. Now, the next step was then the cross-linking and mass spec. So we cross-linked the protein, we digested it, we enriched the cross-linked peptides, they were subjected to mass spec, and we came up with a large set of cross-links. For the core particle alone, there were 71 unique cross-links. We could use the known structure of the core particle to validate the cross-links. and also to set a threshold for what we accepted as a bona fide crosslink. And so we came up also with a long list of distance restraints for the regulatory particle. Now, we did the experiment, as a matter of fact, for two different species, Cerevisiae and Pombe. We used different crosslinkers, different proteases, and so we obtained two interaction maps. As a matter of fact, similar, but by no means fully redundant, as to be expected. And the first thing we could do based on these data was to confirm, the order of the AAA ATPase subunits 1, 2, 6, 3, 4, 5, and more important, it also established the spatial relationship between those subunits and the alpha subunits of the core particle. And again, as indicated here, the central axis, didn't coincide. Then we had to localize the ubiquitin receptors. That was a collaboration with Keiji Tanaka's lab, and so what we did, we used deletion mutants, a deletion mutant for RPN10, a deletion mutant for RPN13, a double deletion or double knockout here, and so we could always, then generate a difference map between the wild type and those deletion mutants, and that, indicated and highlighted here in green where RPN10, RPN13 were located, and the double knockout, very nicely was in agreement with that. Now, this is, I'm afraid, a little bit a busy slide, but nevertheless, let me go through that row here. Again, these are the ingredients of the modeling approach. We had a couple of atomic structures in the meantime of subunits. In other cases, they enabled us to generate homology models. We had the location of the ubiquitin receptors, we had the envelope of the whole thing, and we had the distance restraints coming from the mass spec. So basically, the approach was then the following. Each subunit, or each domain, in the case of multi-domain subunits, was represented in a coarse manner by those beads which were put within the boundaries of the EM density map. Then, these beads were jiggled around, and for each and every configuration, we scored them against the violation of the experimental restraints that came from mass spec. And that gave us, a roughly the position of those subunits. Then the next step was that we used, higher resolution structures or homology models to dock them approximately by rigid body fitting within the boundary of the EM map. And that was then refined by molecular dynamics-based flexible fitting to sort of optimize the EM fit, the crosslinks, and also to the molecular dynamics force field. Now, the result, again, that is unfortunately a bit complicated slide, but the outcome is basically the following, that we evaluated a total of 1.6 million different configurations, of which only 4 shown here, violated either none or at most one of the experimental restraints. So we had an almost unique solution, with the exception of RPN11 and RPN8. These are very similar ones, and we couldn't say which one was which, and a little bit of uncertainty regarding RPN7 and RPN3. But basically, we had now the subunit architecture, RPN1 and RPN2 were located here, at the periphery of the AAA ATPase module. RPN13 and RPN10, at the distal end. The MPN-containing subunits here in the middle, and they were sort of enframed by the 6 different PCI domain subunits. For the PCI domain ones, we suggested already at the time a kind of a quasi or pseudo-atomic model here, because we had obtained in the meantime a crystal structure of RPN6, which we could use also as a template. Also, as you see here, RPN6, like all the other PCI domain subunits, is almost completely alpha helical. The interesting observation was that in the crystal packing, RPN6 formed this kind of hexamer, which had a horseshoe configuration that was mediated by the PCI domains here. And we had observed in the EM structure already this type of horseshoe arrangement. So that was in nice agreement. We could prove that also in a much more definitive manner. We took now the crystal structure of RPN6, low pass filtered it, cross-correlated it with the entire structure of the 26S, and you see here that would be the cross-correlation function. The cross-correlation peaks were extremely sharp and well-defined and confirming the positioning. Simultaneously but completely independently, the group of Eva Nogales and Andreas Martin came up with a structural model for the 26S regulatory particle. And as you see here, there's no need to go into detail. The two models were in perfect agreement, which was quite satisfactory. Now, in the meantime, there had been a couple of developments, technical developments on the EM side, and the most important for us was that, by completely automating the data acquisition, enabling us to operate the microscopes 24 hours a day, 7 days a week, we could obtain a very large data set, still at this point without, the new advanced detectors. So based on a data set that was 10 times as large as the previous one, so a total of almost 4 million individual particles, we obtained a high-resolution map, now a 6 angstrom map here. Now, 6 angstrom is not that meaningful, because as you see here in the color coding, it indicates how resolution varies across the molecules. So it is quite high and better than 6 in the core particle. It's relatively high, also at least 6, if not better, in the ATPase part module here, but extremely variable in regions like this one here, which is RPN1. So, nevertheless, it enabled us then to put forward a pseudo-atomic structure of the whole thing. That is now, for the ATPase part, the interpretation of the density, and that is for all the subunits, the interpretation at an atomic level now, which is pretty good, I would say, for all the PCI-containing subunits and RPN1. RPN2, there was a crystal structure out there from Morris' lab, which fitted quite well. These three, however, were quite unsatisfactory. RPN1, because as you will see later on, it undergoes enormous conformational changes, so the density here is a convolute of very different conformations. And RPN8 and RPN11 are the only subunits which have appreciable amounts of beta sheet, and for that, the resolution was not good enough. So, it was important to refine the structure of RPN8 and RPN11, which we did again by X-ray crystallography, which was augmented or made possible by an auxiliary nanobody in this case here. So that is the heterodimer of RPN11 and RPN2. The interesting thing is that this heterodimer, the recombinant heterodimer, has very low basal deubiquitinating activity. That is the consequence of some autoinhibition, which is, a common feature of many of these cellular proteases. But, that autoinhibition, which comes from this insertion, obstructing access to the active site here, is relieved when, these two subunits are incorporated into the entire complex. Now, this is a movie basically summarizing the structural part of the 26S. So we disassemble the whole thing, and then build it up brick by brick. But I should mention right away, this is not meant to indicate in any way that this has something to do with the assembly pathway. It's just to illustrate the structure. So we dock here the AAA ATPase subunits onto the core. Some has, as you know, we see terminal tails which insert into a pocket, which is related to gate opening. That is a refined structure of RPN1, now docked to the side of the AAA ATPase. That is where RPN2 is sitting. This is now the crystal structure of RPN8 and 11 docked into it. They have an extension which inserts into a very large alpha helical bundle here, and that alpha helical bundle enables what was formerly called the lid, and which embraces the MPN domain subunits to move collectively, as you will see in a minute. And finally, in come the two ubiquitin receptors, which I guess were added relatively late in the evolution of the complex, so they are at the far end of the whole thing. So I think what I will do now is I will just give you a glimpse of the dynamics. So what we have done is we have, using a data set of 4 million particles, performed a very deep classification. Now, I think I might need to repeat that. Basically, that is the result of the classification, and we came to the conclusion to divide it roughly into 3 different conformations. I mean, S1, S2, and S3. I will explain that in a little bit more detail. But you must be aware of one thing, and that is that each of these conformations has still quite some heterogeneity. So we still average over, I mean, an ensemble with some heterogeneity. So what it means is that basically it defines, let's say, the boundaries of the conformational landscape, but I mean we morph here into each other, I mean just the extremes. And it's also clear that, I mean, morphing something into a seemingly logical sequence doesn't mean that we really fully understand the dynamics, but I think it gives you a pretty good feeling. Now we see the same thing from the top, and as you see, the biggest movement is a large rotation of the lid, and also RPN 11 and 8. There's a lot of dynamics here in RPN 1 here, and as we move on, we remove the non-ATPase subunits here to get a better feeling for the conformational changes. I mean, the shift and also the domain movements in the AAA ATPase part. Now, well, one of the consequences of these movements, especially of the AAA domain, is that what was, I mean, under, the conditions which we used first, in which basically ATP hydrolysis was going on, the channel, the translocation channel, or the unfolding channel, if you like, in the AAA ATPase was misaligned with respect to this one here, whereas in another state which can be arrested by, for instance, charging the system with ATP gamma S, they become aligned, facilitating the translocation of substrate. So basically, this is a kind of a model, and we are fully aware that it's still substantially incomplete, or there are parts which are still hypothetical, I have to say. For instance, what is hypothetical is the fact that, we assumed here that, RPN 13 and RPN 10 might, there's weak evidence for that, but no means compelling, might, bind simultaneously to a tetraubiquitin here. And then, in the next step, after, the green part here undergoes the rotation, and, when, the blue part, the ATPase, undergoes the shift, what happens is that basically the substrate and the proximal ubiquitin, are positioned directly above the mouth of the AAA ATPase, and RPN 11 is positioned such that it would be able to cleave this linkage here. I think especially here, I think we have to deconvolute an even larger data set, which will be quite a challenge to get a better feeling of then, the processes will follow. But it will also be essential to be able to stall the process at distinct point in time, because, as I said, morphing something into a seemingly logical sequence is one thing, but to relate it also to, let's say, the biochemical status is a different thing. But given also the advances in technology, and that today with much better detectors we can obtain similar results with quite a bit smaller data sets, makes this absolutely feasible. So my expectation is that in the next 2 or 3 years we will get a fairly complete picture of the events that precede the translocation of substrate into the core. Now, I want to change gears completely and tell you about something which is really work in progress in its very early days. My lab has been interested in, that has been, as a matter of fact, the main activity in my lab over the past 20 years or so, to enable what we like to call doing structural biology in situ. So the idea is that instead of the traditional reductionist approach in structural biology, where cells are taken into pieces, components, fractionated and purified, and then studied in isolation, we would like to be able to study molecular machines in unperturbed cellular environments, which is important, maybe more important for other molecular machines, if you think, for instance, of kinetochores, of centrosomes, etc., which are, I mean, so deeply embedded in cellular environments that it will be impossible to isolate them without affecting their structural integrity. We think that, well, I think many think that entire biochemical pathways are organized certainly in a non-random manner, and it will be important, I mean, to be able to study them by what we like to call visual proteomics. I think it will not be possible to reconstitute such complex environments, including with all the aspects of crowding and so on and so forth. So the method that enables us to do, and which we developed over the past 20, 25 years, is cryo-electron tomography. Basically, what it does is it combines the best possible structural preservation, the best preservation that is physically possible, because the only preparation step is rapid freezing, so that a cell becomes embedded in amorphous ice with the power of three-dimensional imaging, which is, I mean, kind of symbolically shown here for an actin cytoskeleton and, I mean, the ribosomes, etc. Now, it has been a slow development, and there are a couple of problems, especially if you want to study something like the 26S proteasome. One problem is, I mean, we have learned to grow many cells directly on EM grids, and it's easy, well, I should say it's easy to apply cellular tomography to relatively small structures. I mean, prokaryotic cells, or I mean, isolated organelles, etc. That is more or less trivial. The problem is to study intact eukaryotic cells. They are just too thick to be transparent for the electron beam. So, I mean, this is a dictyostelium cell, and you see the periphery is just, I mean, kind of transparent, but the central part where the nucleus is, I mean, becomes intransparent. So the method which we have developed in recent years is called, I mean, focused ion beam milling. It's something that is very common in the material sciences, in the semiconductor industry, but I mean, it had not been applied to amorphous ice. And we were concerned that we might have temperature rises, etc., and this could then, I mean, devitrify the samples, etc. So basically, we use a modified, well, an extensively modified dual beam instrument in which we have, I mean, a scanning electron microscope and a focused ion beam of gallium ions, and the gallium ions are then used to ablate material and to cut kind of windows into a cell. So the whole principle is shown here. I mean, we have the cell sitting here, growing here on the EM grid, and then we have the incoming ion beam, and we ablate material from above and from below. And what is left behind is a thin lamella, which can be between, let's say, 200 nanometers up to half a micrometer or more, whatever we want. And that is supported only by the surrounding ice. So that has then a thickness which is perfectly suitable for electron tomography. We can do that now in a very targeted way by using a correlative approach in which we have developed, I mean, the tools and methods for examining fluorescently labeled cells, I mean, under cryo conditions, and so we can locate, I mean, the features of interest, and we can identify them, and then we can cut with a dual ion beam instrument, I mean, windows or lamella in exactly that region here. I will illustrate that briefly now for something I'm doing currently with my colleague Ulrich Hartl, working on Huntington inclusions, and then that leads over, then I can return to the 26S. So what you see here is basically the grid, the EM grid in a cryofluorescent microscope, and the Huntington inclusions here, they are, I mean, visible, I mean, in green. Now, the next step is that we select now this particular grid here, and so So we have, I mean, this grid here, and we have the cell sitting here, seen now in the scanning electron microscope. And we can superimpose, I mean, the location now of the Huntington inclusion. We can then, I mean, micromachine the window or the lamella for that region, and then we have included in that, I mean, the whole inclusion, which in this case is a fairly large one, measuring something like 5 micrometers or so. So it already fills quite a bit of the cell. So now we can take, I mean, in that inclusion, tomograms of different regions here. It might be a little bit hard to see in the audience, but I mean, you will see all the fibrils. This is still the raw data. There's no denoising, there's no segmentation, nothing. But I mean, it gives you a feeling for what the data look like. This is now at the periphery of the Huntington inclusion here. You see a couple of mitochondria here, which look pretty sick. The same is true for the Golgi apparatus here. I mean, in most cases, there are indications of the severe membrane lesions here. Now, if we go into these regions here outside and take, well, I must add one more thing. There was one very important development. I talked about that in last week's symposium here. That was the development of faceplate, a new way of generating phase contrast and improving contrast in tomograms of this kind, which have, I mean, intrinsically low contrast because ice and protein, I mean, have very similar scattering powers. I mean, we improve the contrast by faceplates quite significantly. That is becoming also now a commercial product. Now, doing that, I mean, we look now into a region, well, very small region of the cytosol, not far away from these Huntington inclusions here. Now, I think it looks at a glance pretty non-spectacular, I guess. You'll probably say we see almost nothing, but it's not quite true. If you look, for instance, that this guy here is magnified here, it's a tomogram of a single 26S proteasome in its cellular context here. So there's another one, I mean, for instance, here. I mean, you see only those which are in a fortunate which are in a fortunate orientation to the well, perpendicular to the electron beam. And again, this is a tomogram of a single 26S proteasome here. And I mean, of course, it's full of them. I mean, there are others where, for instance, which are not double capped. And here comes one of the surprises now. What we can do, we can now computationally extract tomograms of these 26S proteasomes, and we can first create a global average. This is a global average over roughly 1,000 tomograms of single 26S. And as you see here, somewhat surprisingly, I mean, the contrast or the density here in the position of the second regulatory particle is quite a bit lower than it is for the first one here. Now, if you do, again, calculate a variance map, comparing all 1,000 particles in the data set with each other, it explains the situation. The main variance is here. It means basically that, I mean, a large number has no second, I mean, 19S regulatory particle. But on top of that, it's also clear that we have relatively well-defined conformational changes in the second regulatory particle here. Now, we can take that data set and perform then a first classification. And as we do that, the result is that only roughly 1/4 of all the particles in the cell here are double capped with 26S. So obviously, I mean, that is, of course, the preferred thing to study. But I mean, we are certainly biased by the preparation, by the purification protocol, and also in selecting the particles afterwards. The fact is that the vast majority, 3/4 of the particles, are single capped. Now, of course, if you, I mean, form the difference between those two, it's clear that the only difference is the presence of a second cap. Now, as we, I mean, go a little bit further and do now a classification of the single-capped ones here, well, that is again a variance map. Yes, there are conformational differences here, and we can separate them, even though resolution is currently only something like 26, 27 angstroms. But we have analyzed only half the data set, and we expect that we will get to the lower 20s once we have analyzed the full data set. But it's good enough, nevertheless, to discriminate very clearly between two basic conformations. Now, it might be hard to see here the difference, but I mean, you will see it here as we morph the two structures here. So, I mean, there are substantial differences which can easily be picked up at that resolution. And, I mean, we can relate each of the two conformations to, I mean, two of the conformations which we have observed at much higher resolution with single particle analysis. So, well, that is, I mean, the structures docked into them. So, what it tells us is basically, okay, that I had already indicated 3/4 of the molecules are single capped, 1/4 is double capped. Of those which are single capped, roughly 78% are in what we call the ground state, which is basically, I mean, they are ready to accept substrate, but they are not yet engaged with substrate. And 22% only, I mean, are actively, I mean, are in what we call the commitment step. So they are dealing with substrate. Of those, I mean, which are double capped, the situation is, of course, a bit more complex. Roughly half of them, I mean, are again in the ground state. Very few, I mean, only something like 5–6% are in what we call a double commitment step. So they're obviously dealing with substrates at both ends here. Whereas, I mean, the majority of those which are active only at one end. So there's obviously no correlation between the states at the two ends. That is also the reason, by the way, why we have always refused to impose twofold symmetry on 26S reconstructions, because we knew already there from the single particle data that the two ends were usually in different states. So the final one is this one here. I mean, the nice thing is that in addition to being able now to discriminate between conformations in situ, we also keep information about their location in the cell. So we can, for each, I mean, position where we find, I mean, let's say a single capped or a double capped, we can say this one is, I mean, in an active state, actively dealing with substrate, or it's not. So the challenge for the future is quite obviously that we, I mean, work out, I mean, interesting information out of the correlation of positional information and the state of activity, which requires that we scan and examine very large molecular cellular landscapes, which can be done quite systematically, and I think also with the necessary speed in order to come up with some interesting results here. So it's, as I said, this is a very preliminary result, but I think it gives you an idea of the things to come. So that brings me to the end. I should say that is the group of people who worked on the 26S. Florian did all the image processing. Stefan is now a postdoc at UCSF. He has collected many of the data. Andreas Bracher with Ganesh Patara has done all the X-ray studies. Friedrich Förster has done much of the modeling. Eri, who is now back in the lab, worked, I mean, on the positions of the receptors. Pavel worked on the ATP gamma S. Pia is developed, I mean, the classification procedures. And Elizabeth, who is now a faculty at UCSD, was, I mean, the expert for the molecular dynamics, and it was a very productive collaboration with the lab of Andrei Charlie at UCSF, Keiichi Tanaka's lab, and Rudi Ebersold's lab in Zurich. And I should say that is, because I very briefly touched upon that only, is the group that has developed, I mean, the instrumentation and the methodology for the in situ studies by tomography. And I thank you for your attention.
It's a great lecture. I have two quite basic questions. One is, you get a quite clear structure for 26 adipolysome, and also get a clear process for binding to ubiquitinated protein and degradation. So where the end product, peptide, come out from 20S, so tight structure?
Yeah, that is a question, a long-standing question where it really leaves. I mean, one could now say, of course, that it's single-capped. I mean, maybe the other exit is available, but I think it could be similar to indications, let's say, coming from, for instance, NMR studies in Louis Kay's lab with the 20S alone. I mean, that also there might just be enough dynamics also in the core particle to open up, I mean, for a short time in between the beta or the alpha subunits. I think the latter is the more likely explanation. I think we tried to address that experimentally. We tried to engineer using, I mean, well, cysteine mutations, cross-linked ones, which would not be able to undergo this type of breathing in the core particle. But I mean, unfortunately, these studies were not, didn't lead to conclusive results. But I guess that would be my expectation, that it opens up temporarily.
Yeah, also, I think I'm extremely impressed by your in situ progress for the 26 proteasome.
Pardon?
The in situ in the cells, you can look at the proteasome, 26 proteasome specifically. It's quite amazing. So I was so surprised because unlike the X-ray, you can penetrate well. Mm-hmm. So for the EM, it's hard to look at it in depth, right? The inside.
Yeah. I mean, well, I mean that the picture might be misleading. We looked at it, of course, in the entire volume that was enclosed in the tomogram. So I mean, of course, this was in the end a two-dimensional picture, but I mean, we looked into the entire volume. And I should say, these studies were designed to look at the aggregates and not at 26S. So if the goal had been to study the 26S, we would have used a different magnification, a different pixel size. So as I indicated, so we think that with the existing data set, which is already a fairly large one, we will get probably to something like 20, 22 angstroms. We are very optimistic that by optimizing the parameters, we can also in cells go to 1 nanometer.
Yeah, I think maybe it's easier to do in the cytosolic, for cytosolic protein. Also, proteasome mostly should be in the nucleus. So have we ever tried to look at
we have, I mean, no, we have, for the sake of simplicity, currently looked only for 26S in the cytosol. It's a little bit easier because it's less crowded there. So, I mean, it was
get a DNA and everything else there.
The nucleus is terribly crowded. It has a higher density also because of all the DNA and RNA there. But I mean, it will be possible, but again, I think the experimental conditions would need to be modified. We would need to go to relatively thin slices or thin lamella in order to, but currently we are happy that we can look at them in the cytosol.
Yeah, I have a curious question. So you I mean, this is so cool, the proteasome in situ detected by the ECT. So I'm just curious, all these different forms, ground state or committed or double cap or single cap, are they just simply in the soup, or are they actually hanging on to some filament or tubules or some solid support?
No, I mean, we have not systematically looked for that. I mean, some are relatively close to microtubuli, but I mean, they are basically, well, maybe this has a little bit to do with the way they were selected, which was done in an automated way. I mean, we used, for cross-correlation, a 26S double-capped template to perform the search, but it easily picked up also the single-capped ones. But it might be that needs closer inspection, that a 26S that is, for instance, directly associated with, let's say, an actin filament, or let's say with a membrane, is not as easily detected as one that is free-floating in the soup of the cytosol, as you said. Okay, so we would not at this point in time, make any sort of statements about, let's say, the systematics of interaction or cellular distribution. I mean, that is a little bit more complex. Yeah. Dan.
With the new detectors, what do you anticipate as the practical limit of resolution? I mean, would you be able to reach like a trip channel-like resolution, or do you think there's just too much inherent heterogeneity
now,
in these
in
probes?
single particle analysis or in cellular tomography?
No, single particle.
In single particle, I think I don't see any reason, if working hard, that I think 3.5 angstroms is absolutely feasible. I think it will be the easier, the better the conditions are defined, stalling the thing in one or in a limited number of conformations. I mean, as long as we have the whole range of conformations, I mean, you must basically multiply the number with the different conformational states, and each conformation has to be sufficiently populated. But by roughly, I would say, with the new detectors, roughly, I think a subpopulation for averaging can be 10 times smaller as it used to be, so it's becoming feasible. In cellular tomography, I mean, the detectors are a little bit less important than the faceplate, which, I mean, improves contrast so dramatically. So we never saw 26 as clearly, even with a new detector, but with, I mean, enhancing phase contrast, they really pop into the eye.
Just because you gave such clear percentages in the cell which proteasomes are double capped and which just have a single one, did you ever test several stress conditions to see whether this changes?
Sorry, I didn't get the second part. Single capped and double capped in the cell?
Yeah, and did you check several stress conditions to see whether this changes? If you have, I don't know, rapid upregulation of misfolded proteins in the cytosol.
I'm not quite sure I fully understood your question. Maybe we can discuss it afterwards. I think we see the same conformational states in the single capped and in the double capped. I mean, we don't see any difference beyond, I mean, as a matter of fact, even if you, I think the fraction that is in one or the other conformational ground state versus what we call currently commitment step, we think, by the way, that with the complete data set, we will be able to discriminate between all three basic conformations. But we don't see any difference there. We don't see any difference at that resolution that is sort of unique to one or the other form.
Okay, have you been able to correlate the different states of RPN1 to any other parameters systematically?
No, not yet. I mean, what you saw in that movie was, I mean, the result of basically a focused classification on RPN1, so that, I mean, we deconvoluted the coexisting conformations. That can be done very nicely. I mean, I think we would not be able to say at this moment, I mean, how it correlates with the other conformational changes. We are also working hard on the interaction of RPN1 with UBP6, which is, I mean, an obvious thing to do. It's very difficult. I mean, that might explain a couple of things, to have it with UBP6 bound. It is very difficult to optimize the occupancy with UBP6. I mean, it's usually quite substoichiometric, but there is, that is currently Aries project. There is progress there. I think that might, but if you mean do the relatively large conformational changes of RPN1 correlate with additional, let's say, density that would come from, I don't see that yet.
Yeah, I mean, like motions of the lid,
Yeah.
or the different states of the ATPases.
Yeah. No, I mean, it's just, I mean, currently it basically we can, after classification, make it fit quite well to to the EM density. Yeah. But
so is RPN13 a permanent member of all the proteasomes in situ?
no, it varies enormously in stoichiometry. I mean, for instance, I think there is relatively poor occupancy with RPN13 in Pombe. It's quite a bit better in Cerevisiae, but I mean, we usually, and that is why it looks a little bit loosely attached, I mean, because the density shrinks a bit. So the better data are obtained with Cerevisiae, but I mean, if you look, and we have done quite a bit of quantitative mass spec, it's usually, I mean, an occupancy of between 10 and 30% in our hands with the systems we are working with. So it is, let's say, an intermediate between a resident and a kind of a shuttling ubiquitin receptor.
Okay. Any other questions? All right, good. Oh.
In your in situ studies, where you were using the Huntington fragment, did you see the HTT co-localize with the 26S proteasome?
No, but again, the in situ studies of the 26S were a kind of a byproduct, and I mean, the Huntington project is just in its very beginnings. I mean, we are glad now that we have cells which, I mean, we can easily grow on a grid which express, I mean, well, it's a very long molecule repeat. We are glad now that we are able to target them precisely, to cut the lamellae and to examine them, but we have neither analyzed the fibers as such, nor have we analyzed the molecular inhabitants. Currently, it's an ongoing study in which my colleague Matthias Mann is doing the proteomic studies of the inclusions, and we are trying to, I mean, to get a structure of the inclusions. But it's too early days. I mean, we see many things, but I think we see mostly that at the periphery, basically all cell organelles look pretty sick. And it's also, I mean, those areas are fairly densely populated with 26S. But so far, as a matter of fact, I have not seen any 26S, I mean, really deep in the inclusions.
So in your situ study, you see the majority of the 20S is singly capped, and also only 24% are doubly capped. So it's like in nature, is singly capped is enough to perform the...
Obviously, yes. I mean, that is, I mean, it came as a surprise to us, but because, I mean, we also, in all our structural studies, of course, selected the double capped for the structural studies, and I guess also, I mean, the purification protocols are sort of maybe geared towards, I mean, optimizing the yield of 26S. I mean, well, Dan has a long time ago tried to discriminate between single cap and double cap by native gel electrophoresis. And I mean, they coexisted, but I don't think, well, you would not claim that these studies were quantitative in any way, because, I mean, they are. But we always thought in the past, I mean, when we saw the single cap, yes, they have just fallen apart. I mean, that was a kind of an assumption. And so the image was always that of a double cap, but I mean, that is definitely not true if you look now at the cellular situation. And I think they must, well, I think the fact that, I mean, we can correlate them with the existing conformations as they go through their functional cycle, I mean, means that they must be functional. And I mean, I think it's also, I mean, I think, again, I think going back to Dan's old experiments, you would also say that the bands in the native gels, which have single capped ones, are proteolytically active, right? Yeah. So
then
it's not
one last question. Why,
yeah.
I mean, then why the meaning of the 26 as the doubly capped?
Why is it? Well, I mean, I think it could well be that, I mean, for instance, let's say it could be in some ways reminiscent of the, again, also different from the situation with GROW-EL, GROW-ES, where, I mean, I mean, the caps might alternate between the two ends, okay? And so, I mean, it is a special situation that, I mean, they exist at both ends.
Okay, good. Thanks. And thank you, Wolfgang, once again. So